My name's Carl Edwards. I don't have any slides because this is going to be slightly random. Anybody who's met me before probably will be unsurprised by this. My background is I had a real job once. I was a scientist, microbiologist, and then in the renal transplant unit. Due to the vagaries of academic funding, I drifted into commercialisation. So consultant to venture capitalists, floating companies on stock markets. And you kind of see almost everything from the really slick, well-planned startup to the, we have a good idea and we got £1 million, what are we going to do? So it's an interesting apprenticeship doing consulting. I decided after doing a substantial amount of consulting on clinical trials in particular, that being an ethics committee member, and then I became an ethics committee chair, it's poacher turn gamekeeper. You can kind of read into what they're trying to achieve, and maybe some clinical trials weren't being done for the best possible reasons, financial rather than clinical. And I did that for 15 years. So we specialised in two committees, Leicester and Nottingham. Leicester specialised in new drugs, new chemical entities. We did lots of the paediatric trials as well. The Nottingham one had Mental Capacity Act trials coming to it. So researching with people who were unable to consent through various reasons, and kind of doing that sort of background. And interwoven with that is kind of the day job, which turned into commercialisation, university tech transfer. And I guess the kind of my view of sustainability, we can look at like sustainable packaging or sustainable energy, but actually there are other things that need to work. And if you don't get them right first time, the thread runs through a product or runs through a company until it finally stops you. So if we look at, say, design for manufacture, let's throw back to Lucy's example of the device that works, but actually it's encapsulated, it's not recyclable. You have to sterilize it in a very particular way that's really expensive, or it uses materials like angel's tears that are really expensive, which is fine for a prototype, but when you want to make $1 million, what are you going to do about that? And taking that advice on design, I mean, locally we have a number of very, very good design houses. I work a lot with a company called Renfrew, and they will help you and have helps me, as well as my academics, think through, okay, this is what you want to do, but are you really sure you want to build it out of this? Because later on, you're not going to be able to buy enough of it. There's only so much of this in the world. And I'll come on to that later on. The other thing about working in healthcare is everything is regulated. Everything. Everything has a set of rules, from a tongue depressor through to an implantable defibrillator. There's a set of rules. And there's a limit to about the extent to which you can deviate from them. They're risk-based rule systems for devices and diagnostics. I don't know, people more interested in drugs or diagnostics or just interested in healthcare in general? Yeah. Yeah. But the thing is, when you're looking at drug development and drug regulation, it's quite regimented. People know what to look for. In devices and diagnostics, it's risk-based. There are a set of rules for can you produce a tongue depressor, but they're much shorter than the ones for, implantable defibrillator. Same with in vitro diagnostics. If you've got a diagnostic test which is non-critical, say for hemoglobin or something like that, it's done many, many times, and if you get one wrong once, that's fine. If you're doing blood typing, the risk appetite for getting your blood typing test wrong is 0. And there's a whole set of different rules, and they ramp up, and how you test them affects what you do. And that might B, you have a vision for your company that says, we are going to be paperless, or we aren't going to do any animal testing. And you run down the road and actually the regulator goes, see those monkeys over there? You're going to inject your drug into them. Now, this could have been planned for, but now you've run up against, you haven't planned ahead. I'm a great believer in not having any surprises, because the surprises are always the things that are horrible. So if you look at testing, let's look at testing. You might decide, okay, I want to be animal-free. Plan it, talk to people. There's an entire industry and charity sector set up to help people avoid using animals in testing. There are two really good reasons to avoid animals in testing. One, mice are not furry test tubes. They are living things and shouldn't just be used as furry test tubes. The other thing is they're really expensive. Anybody who thinks that anybody does animal testing because of the joy of it really hasn't looked at the bill. I was involved in a project in Nottingham where we were looking at a synthetic oxygen carrier, injectable. And to do that, we were looking at novel detergents. And these detergents had to be biocompatible. Biocompatible detergents that you can inject. Yeah, that's a steep learning curve. So how do you test them? Do you inject rats with them all the time? I kind of went to our local frame, that was based in Nottingham, Funds for Replacements of Animal in Medical Experimentation, and said, I have these detergents to test, what do you suggest? And they said, look, here's a couple of tests, you could screen them and get rid of the ones that are actually going to be quite dangerous. And that then builds into your development pipeline for the people who would have come after me. Actually, we got a really high throughput screen is the wrong word in the sense of drug development, but it's a high throughput way of screening out the compounds that are just not going to work. So you can concentrate on the ones that are going to be user-friendly. I can't read my own handwriting. Arms aren't long enough. So yeah, oddly enough, that was I can't read my writing. So another one is, is your method of testing sustainable? And I'm going to use a really good example of people use a particular test for checking endotoxin content in preparations, drugs, injectables. Limulus amoeba assay. Sounds benign, except it requires you to find horseshoe crabs and line them up in a lab and milk them. Currently, there is no real accepted alternative to this. So if you don't like milking limulus horseshoe crabs, then you can't produce your drugs. So, there's things like that, if you've got that statement in your, we are not going to use any animals at all, at some point you might stumble across, actually, we didn't realise this. And that's oddly enough, it's a real problem, because there's lots of injectable preps and only so many horseshoe crabs in the world. And because the regulatory bodies are, well, this is the test we would like, it becomes an interesting kind of hamster wheel to get on as a company. You've also then got to look at human tissues. Are we going to experiment on human tissues? Certainly better than using animal tissues, but how do you source them? Any idea how if you are producing an antibody, you have to check that there's no non-specific antibody binding to any organs. How do you think we do that? Yeah. How do you harvest brain tissue from healthy individuals? And it's this kind of can of worms. You lift the lid off and go, oh, well, actually, that means healthy individuals, we have to have liver, pancreas, brain, spinal column, bones, which essentially means they really have to be dead. And that means heading this off by having a whole set of donors who are healthy lined up before they die. Sustainable? Well, there's lots of people.
They agreed.
Yeah, that's the thing. You have to find somebody to agree to it. I was involved in a project that actually did that as part of the ethics committee. So people who were healthy, or at least mostly healthy, could line themselves up when they died to have particular samples taken before a post-mortem, or if they weren't having a post-mortem, kind of before they were given over to the undertakers. And it's time critical as well. Somebody has to know this and get them to the right place. So there are things that, again, no surprises. Anybody who was thinking of developing an antibody, that's your next surprise. There's also then, okay, how are we going to test a treatment or a device? If you've got something relatively benign like a painkiller, then there's a well-trodden route. You can kind of follow in the footsteps of paracetamol, like ibuprofen, tramadol. But what if you want to have a treatment for some people who are critically ill? Say something you would give somebody in an emergency department. They can't consent. How do you do research with people who haven't consented to it? You have to anticipate that. You know, if you're thinking, right, what I'm going to have is a drug that stops brain injury after a severe trauma, the only people you are going to test that on are people with severe brain injury. Ingrowing toenails just won't cut it. And this would my ethics committee hat on. And ethics committees, whilst they have quite a bad rep, are there to help people help people. Ill people need therapies. Toru's particular speciality is aortic dissection, where your aorta out of your heart bursts and then you have to do something about it. You can't check your therapies on anybody who doesn't have this condition and who isn't in a lot of danger. How do you do that ethically, and how do you get these patients on board? And you need clinicians like Toru on board if you're a company to say, right, I'm thinking of doing this, and they'll go, you have no chance. That's too dangerous. We won't leave people that long. And that, it's sustainable planning, no surprises. If I look at kind of the regulations and kind of how you test it, there's a risk of getting it wrong, but also there's a risk of getting it right. So I'm going to go for two diagnostics because the diagnostic ones are the kind of easiest ones. So at the start of the pandemic, everybody scuttled off. We all had sequences. Right, we got a PCR test, and locally we had a LAMP test that would test for COVID. Great. What happens if we don't detect it and somebody is positive when we're testing it? What are the consequences? And you have that's the kind of thing you have to think about before you even start. And the decision was that, you know, our testing was just screening. We couldn't offer a diagnosis with our level of test, but it was indicative. Equally, if you are a diagnostics company, quite often you will want to try out your diagnostics on a healthy population to ensure that you're not giving false positives, particularly for quite sensitive tests. Quite a lot of companies have panels of people, we're back to volunteers again, who will have blood samples taken or urine taken or whatever as their negative controls. What happens if you've got a HIV test that suddenly detects somebody with HIV who's supposed to be healthy? This is a real one, by the way. And the company's like, we haven't actually got a protocol for this. How do you get there? How do you test healthy people for HIV and not have a protocol for if somebody turns up positive? I mean, this is really kind of on the far end of surprises you really don't want. It took a lot of unpicking because one of the things is they promise never to contact their patients or to contact their participants about the test results. Lack of forward planning. But again, it's that thinking ahead, how are we going to keep the business going in the long term? And that would have just required a little bit of planning. Equally, you can look at supply of medicines and devices. So if you are doing medical research, if you have a group of participants in your trial and they benefit from your treatment, can you withdraw it at the end of the trial? So say your company decides actually, we're going to finish that trial and it's not going to make that much money. Can you just stop giving them that drug?
Depends.
Not really. Declaration of Helsinki says you keep treating them, whether you're producing it for a profit or not. Once you've participated in a clinical trial, you are a special person and deserving a special treatment. So I've got kind of 3 examples that kind of rattle down the kind of possible consequences of that one tiny rule. We had a company that had produced a batch of endothelial growth factor, incredibly expensive, but had only produced 1 batch and basically gone bust. But they had a batch of clinically available endothelial growth factor, and a clinician wanted just to try it out in people with ulcerative colitis and actually treat them with endothelial growth factor to see if this treatment would work. Of course, the one thing you can't do is offer anybody this after the trial. And so is it ethical to have these people put themselves to the hazard, to borrow the jousting term, to check whether it works to see if somebody else wants to make it? Would you let people do that? Show of hands. Who thinks, yes, let's try out some EGF on people with ulcerative colitis?
Did you say it's been proved to be successful?
No. Literally, this is a suck it and see. They have a bucket full of EGF that's generally regarded as safe. It's a protein. It's not a small molecule. That's why it's incredibly expensive. Show of hands. Yes. Do we do it?
I'll go yes, because no one's saying a trick question.
The conclusion was yes. Anybody know anybody with ulcerative colitis? It's not a nice disease. The side effects of EGF are next to nothing. It's like crack on. It didn't have a huge effect, certainly not worth anybody picking it up, which is a shame. But you could have easily seen, roll the dice, actually this is a wonderful therapy. Suddenly it's worth it, because if you've got ulcerative colitis, your end game is high dose steroids, horrible drugs, surgery, death. So actually, that was the ethics committee, kind of 14 odd people, all sat around and went, yeah, let's do that. Equally, you've also got the regulator gets involved in this. So if you're a drug company and you've produced a drug and decided actually it's off patent, I'm not making very much money out of it, I'm just going to stop making it. Hands up who thinks the regulatory authorities can still force you to supply it. Yeah. GSK had a pretty average antihistamine called acrovastine. And they wanted to withdraw it from the market, and the regulator said, no, you still have to produce it. GSK really didn't want to do it, and they outsourced it to a company in Northumberland because the synthesis of it is horrible. But again, law of unintended consequences. If you don't plan ahead, if you don't realise these things can happen to you, you could end up supplying a dead-end product that costs a fortune to produce, which it does, into a market that's buying something else. But a small number of people wanted acrovastine and they were stuck. Locally, this was a really good one. Again, a drug trial of a new, back then it was brand spanking new diabetes therapy, but it was much easier to manage than insulin in people with type 2 diabetes. And they decided to try and do this during Ramadan. Whilst people with diabetes are encouraged not to fast during Ramadan, it's an act of faith. People are perfectly entitled to do it, but how do you help them do it safely? And this particular drug was spectacularly successful. These guys their blood sugar was fine, nobody collapsed, nobody was admitted, it was like off the scale as far as the slight positive effects they were looking for. And obviously these guys are then discharged back into the community and now want the drug. And the local funder was like, oh, well, no, we're not giving these people this drug. And it's like, I'm sorry, guys, you have to. Because these people have put themselves to the hazard. And as a supplier of that drug, you are then, the supply is not too bad, but that has a massive impact in the NHS because you're now actually buying a relatively expensive drug. It didn't break anybody's bank. But the NHS now has an impact and hasn't really thought this through. I've got some real horror stories. These are the good ones. And I suppose sustainability, I'm going to wrap up on one that's quite popular. AstraZeneca vaccine. Is anybody still using it? Worldwide? Does anybody know?
I think they probably are supplying it still.
Yeah. It is by far one of the most widely available vaccines. Easy to produce, cheap to produce, easy to transport. Almost everything that the mRNA vaccines aren't. It has really good efficacy. It has a tiny risk of stroke, which we can come back to, which again falls under the law of unintended consequences. But that was the result of a vaccine program that was designed to do something else, really. It was for rapid response to tropical diseases, rare diseases. And it was an IKEA virus that they then put the sequence in, produced it, and got a very effective vaccine. Hands up, who had the AstraZeneca vaccine? Yeah. But there was evidence that it did cause stroke in some people. Did anybody read up on what had happened? Everybody, at the time it was like, vaccines don't do this. There is no reason a spike protein would cause people to have a stroke. And actually, law of unintended consequences. The viral particles you produce as a vaccine are negatively charged. If they leak into the bloodstream, they interact with your platelets, and in some people, they gather the platelets around them and kind of create a runaway kind of gathering cascade that creates particles that are just the right size to pool in this one blood vessel in your head. You really couldn't make it up. And so it's nothing to do with the vaccine, it's the actual viral particles themselves. So they can actually engineer that out for the next version. But again, because it hadn't been tried before, the first time anybody found this out was when you inject millions of people. Whereas if they'd have tried it prior to this with smaller vaccine numbers and smaller numbers of people, it would have been picked up that it caused this kind of platelet depletion. But, we don't hear a lot about it, but it's made by the Serum Institute of India, one of the world's largest vaccine producers. It's dead easy to ship out, really effective, used all over the world, but we don't hear about it. Is it sustainable? Absolutely. Are mRNA vaccines sustainable? There's a lot of money needed to make them. There's a lot of investment, and you can we keep throwing money like that at mRNA vaccines for infectious diseases? For cancer? Maybe not, but infectious diseases is a lot of people. And that's my mazy dribble through kind of no surprises. Any questions?
Any questions or comment? Anyone? If there's one take home that you'd say, Carl, I know it's sort of preparing for the is.
It's never what you think. The thing that trips you up on a wet Tuesday, it's never what you think. Which, interestingly, I am not the world's greatest planner. I generally have a plan, but I always generally know it's not going to work. But you have to have a plan. And that is the first, that is your first step. It's like, okay, my first question is, has anybody else done this before? Ask them. I make, for my sins, I've got a hobby, I make costumes, armour out of floor mats, rubber mats. You shape them with a heat gun, you coat them so they can take paint and glue them together. But how did I learn to do that? Well, I watched YouTube and I asked other people who did it. Now I'm in a position where other people doing it ask me how to do it. And roughly, that's where the ethics committee job comes in. I've seen lots of it and people go, Carl, you ever seen that? I go, yeah, seen one of those, don't do that.
That's why we used to select you as a researcher. This was our favourite chair to come to because he was so pragmatic. And yeah, and if we were allocated somebody else to chair,
But I'm old, so therefore I am wise.
So, that's the question. Yeah. So what do you think would close the gap for a young company who's got this great idea but hasn't got the old and learned and wise stage? Because often they won't talk to anybody else because it's and one of the things we do in our innovation surgeries is we say we do not want to talk about ethical protection. We don't want to see the actual design, but you can tell us what it roughly is.
Yeah.
So what do you think? What do you think we should try and put on as an additional thing for the Legion?
But the interaction with the people who actually do the job. So, you might have a great idea for a cardiac intervention, but the person who's going to be using it, it might work, and you go, well, that's lovely, but I'd never use it. And that does happen. Real life example. Company got up to the stage of getting an MRSA assay that would work in 30 minutes. A bedside assay, MRSA. This is in the early 2000s, so it was dead popular. But hadn't talked to any clinical microbiologists. And as a consultant, that was my job. I went and talked to some clinical microbiologists. I am a microbiologist as well. And the clinical microbiologist went, no, I wouldn't use it. Why not? And here we are, the law of unintended consequences. Well, it's going to take 30 minutes. Yeah. I'm only with that patient for 7 or 8 minutes. I need to make a decision whether to put them on vancomycin there and then. By the time I get back to seeing them, we'll have a result back from the lab. And we'll know if it was either right or wrong. And you're like, you are kidding me. I went, nope. And I spoke to 15 different clinical microbiologists because I really didn't believe the first one. And they all said exactly the same thing. The timing of it. 30 minutes was still 23 minutes too long. Because if you do have MRSA sepsis, they have to do something right there. And they're not going to be back for a while. So getting people who do the job involved and knowing what's involved, that's, I mean, as I said, people come to me because I've done the ethics committee thing, because I've seen lots of people come through with good ideas, bad ideas, things that works, things that didn't, because they always have, everybody who does a clinical trial or whatever, the results always come back to the committee. And one of my jobs was to read back, we approved this, didn't work. We approved this, was a tremendous success. And, that it's a learning thing. And also it's satisfying that, the committee members don't get paid or anything, to know that, you contributed to how diseases were treated worldwide. It was like, that's worth the tea and biscuit. But yeah, it's that interaction with people who do the job. Sorry, you're the only clinician in the room, so.
So, Tara, then, if if a company comes to you, and this is a hypothetical, obviously they've got into front door to the LLSA or somebody who knows you and can contact you. What would be the things that you would be looking for that would give you faith in interacting with that company? Because obviously NHS is a nightmare at the moment, really busy. What would make you, if you were a company coming along, that would make you kind of go, actually I could give them 10 minutes to think about this?
I mean, as Carl mentioned right now, it's that unmet need. It's how much impact they would have, that gap, clinical gap, I guess, if that makes sense. Because, you know, if they go for a very rare disease that nobody else has gone for, you'd probably give them more than 10 minutes.
Yeah. How would a company or those who are thinking about developing stuff know what the unmet need is? There's not a list out there that says unmet clinical needs, is there?
No, but there are a lot of patient groups out there nowadays that advocate for that unmet need. I think one of the big differences in the last decade or more are, in a good way, the vocal patient groups who are advocating not just to us, but they work their magic with the politicians too, in ways that we don't know as clinicians. I've found that out. So, you know, those people there's strength in numbers when they come together, if that makes sense.
Yeah.
So, would you then think that doing something that brought together designers, entrepreneurs, patient groups, and some clinicians would be a good way of looking at? So we've obviously got massive health inequality in Leicester. Are there things driven by unmet clinical need within that population?
Yes. Now, if you're able to answer something and the public is behind it, and clinicians are willing to support it, I think, you know, you have a chance. What do you think, Carl?
Well, the thing is, it's almost like it's a huge elephant you're trying to eat. So, but kind of years ago, we did a very small thing, but it was a program. Wouldn't it be great if? So what we did was grab a load of respiratory guys, actually, Mick Steiner included, and bring them over to Loughborough and trap them in a room with a load of engineers and designers. And it was, you know, okay, if you had to turn up, wouldn't it be great if on Monday I could do this? And, you know, there were various things about knowing people's blood oxygen properly, or actually CO2, you know, having that ability to remotely diagnose somebody without actually having to drag them out of the house, having a memory. You know, this is a few years ago, but it's quite a small part of the elephant, but it's quite manageable. I mean, in that case, we didn't have patient advocates in that room, but we had the community respiratory nurses who more than adequately kind of stood up and said, look, you know, this is what worries these people, these guys are in. But wouldn't it be great if you went into work next Monday, if there's one thing you would like to be able to do that you can't do today, what would it be? And is that a soluble problem? And that little event, it's spawned a few projects, but one of them was Mick Steiner working with the exercise guys from Loughborough to come up with less painful ways of exercising. Because exercises come in all shapes and sizes, and elite athletes do very particular things for very particular muscle groups, whereas the previous prescription for exercise for people with obstructed lungs and stuff like that was just go for a run or go for a walk or cycle on a bike. And they're like, oh God, really? and there was a whole program about how do you make that easier and less painful, and people will do it more. You know, if that was one thing that came out of that event, it's like tick Fiji.
Easy exercise really go together, doesn't it?
Yeah, but the thing is,
The whole idea is meant to be hard, isn't it?
Well, no, it's not meant to, but it doesn't have to be painful to be effective.
No.
And that was the conversation Mick had with the exercise guys, and that led to the whole, well, can we come up with things that are less painful? You know, things you can do sat down that do the same as, you know.
Low calorie beer would be.
Yes, so
Well, it already exists.
I was about to say I don't rent this by the hour, mate. But yeah, and that, you know, get it. It's great having a treatment, but if it's so painful, patients don't use it. You know, it's pointless having it. You know, we got a project at the moment about kind of exercise outside in kind of potentially polluting environments. How do you kind of guide people away from where there's high pollen, high dust, high PM10s or whatever, you know, using satellite data, you know, just to make that walk just slightly easier and slightly less damaging. I mean, currently, I get hay fever, so currently outside exercise for me, it's not happening.
I think just coming on to Carl and Lucy's point about the unmet need, I've actually worked in the past on a sort of, it's like health campaigns. So I worked on it was actually headed up by a pharmaceutical company that was in essence bankrolling the campaign, but actually worked with a number of patient groups and charities. It was called the Harvard campaign, and it was to actually reduce the number of sort of undiagnosed HIV/AIDS patients in generally men who have sex with men, but also a couple of other groups, because actually they were quite underserved. But they actually used it was Dean Street down London was actually sort of the main sort of focus of where they were actually doing the treatment and a lot of the work. So it started within London, but then it sort of went out sort of nationally. So I think to sort of define the unmet need, it's actually coming together with the charities and the patient groups and actually building patient advocates to sort of work with those. And then also, I think as well, it's having a look at how sort of interventions or sort of diagnoses are appraised. So sometimes it's, I don't want to go into it now, but there's a type 1 or type 2 error of how things are appraised, and actually how the COVID vaccines were approved was actually a type 2 error, because actually what would happen if we didn't put that intervention in, whereas generally it's a type 1 appraisal. So there's quite interesting things, and actually if you're developing a value proposition, if you can show that you're actually covering both angles, that you're defining a clear unmet need, but it's also efficacious and safe, but that's something on the side.